Hello and welcome to another one of the TwinsUK 30th anniversary webinars. My name is Chris Hammond and I have been working with TwinsUK, oh gosh, since 1998. So that's many years, not quite the 30 years, but certainly a long time. And I'm the Professor of Ophthalmology, that's eyes, at King's College London, working with the TwinsUK, leading the eye studies within the Twins. And it's a joy to talk to you all. And today's subject is going to be about genetics. And I'm very happy to be joined by my colleague, Emma, who will introduce herself to you.
Hello, everybody. Again, I'd echo Chris's comments to welcome you to our seminar today. My name's Emma Duncan, and I've been working with TwinsUK as my main place of work from the middle of the pandemic, because this is really the best time to move internationally, as you all know. But I first started to work with the Twins in terms of collaboration back in the early 2000s or so. So I'm a professor of clinical endocrinology. Endocrinology is the study of hormones. And within that, I particularly work mainly in bone and neuroendocrine tumors, but I've worked with genetics of many endocrine disorders over the years, both single gene disorders and complex disorders, which we'll be talking about today.
Thanks, Emma. And I guess, Emma, maybe we could kick off. We've had questions from the Twins that we've been asked to answer. And one of the Twins questions is, so what is different about the genetics of twins? Why do we study genetics using twins?
Oh, thanks, Chris. So the first thing I thought we'd do is just sort of say, well, really, we talk about two types of twins, generally speaking. We talk about monozygotic and we talk about dizygotic. Zygote is a fertilized egg where an egg and a sperm have come together. And so a monozygotic twin pair would be where both twins have come from one single fertilized egg, which has split at some stage in development. Whereas dizygotic twins have come from two separate fertilized eggs. And so this means that the monozygotic twins share their genetics more or less absolutely, because they've got the same genes as that original egg and sperm coming together. Whereas the DZ twins each have the same similarity as you'd see with any other sibling pair, brother-brother, sister-brother, sister-sister. Those are slight assumptions about the absoluteness, because even monozygotic twins are not 100% alike. So for example, fingerprints are not exactly the same, as even identical twins, monozygotic twins, touch different parts of the placenta and different parts of the uterus as they're in utero. And dizygotic twins are a little bit more similar than normal brothers and sisters, probably because you need to be a little bit more similar to be able to tolerate that. But as an assumption, monozygotic twins are essentially 100% the same genetically, whereas dizygotic twins are on average 50%, the same as any other brother or sister pair. The difference between twins, though, and other brothers and sisters is assumptions about environment, because what is difference between twins and other brothers and sisters is that they've been in the uterus together, and they've often grown up at the same time in the same household. And so what makes twins so interesting is that the monozygotic twins and the dizygotic twins share their environment very similarly, although genetically, one pair, the monozygotic twins, are identical, 100% sharing, whereas the dizygotic twins are 50% sharing. And this enables you to do a whole lot of statistical modeling about discovering the genetics of diseases.
Thanks, Emma. And I guess that kind of leads us, talking of modeling, onto the heritability of diseases. And obviously, I think Tim has discussed that a bit in his first webinar with Debbie, but maybe just you could remind us about what the heritability is and how it helps us sort out genes and environment.
Oh, absolutely. So if one thinks about any feature of a person, and we often talk about this as either traits or phenotypes, whether that's eye color or height or body mass index or weight or bone density, which I'm particularly interested in, or various other things, we have a spread in the population. And if we could think about height, for an example, we'll have an average height, and 50% of the population will be taller than average, and 50% of people will be shorter than average, and there will be a distribution in the population. we describe that distribution as variance. And what heritability is to say, well, with that variation in the population, how much of that is due to genetics as a proportion to all of the variation overall? So heritability from a maths perspective is the proportion of the total variance in the population, which is explained by genetic variance. And so if you're looking at twins, you can do this by looking at the correlation of how similar monozygotic twins are, who, as we've mentioned, share 100% of their genes, compared to the correlation of these phenotypes or traits in dizygotic twins who share on average 50%. And then you can do maths to work out how much of a particular disease is heritable, how much of the chance of getting a disease or the spread of a trait are due to genetics for that particular disease. And this is very useful. So you can have conditions which are 100% heritable, which means if we knew what all of the genes were, we'd be able to predict who was going to get this. We would be able to use that absolutely. So an example of that might be, for example, ankylosing spondylitis, where it's almost entirely, whether you get this or whether you don't get this, it's almost entirely dependent on your genes. I'll just emphasize that doesn't mean that the environment isn't important. It just means that the environment doesn't contribute to the variation in susceptibility in the population. In contrast, being hit by lightning, which is almost entirely an environmental thing, like standing in a field when there's a thunderstorm going on, that would not be a very heritable trait, and you would not find much underneath the genetic, but unless it's the genes for foolhardiness, perhaps.
Good point there. One of the questions we've been asked to answer by the twins is about same-sex and different-sex twins. And maybe, Emma, you could just, obviously for our twin cohort, TwinsUK, we've largely used same-sex twins almost exclusively. Why is that?
So I think that there's several reasons for this. So the first thing I was going to say, Chris, is that the TwinsUK cohort has some unique features. As a general comment for twins in the population, about a third of them will be monozygotic and about 2/3 will be dizygotic. However, when you've got a collection of volunteers, we tend to be enriched more with identical twins. So we're a little unusual. We have more identical twins than we have dizygotic twins than you'd predict by chance. And if I could just stop and digress for a moment, I think, so the chances of having monozygotic twins is uniform around the world, because the chance of a fertilized egg splitting is uniform. It doesn't matter which nationality or ethnicity you come from, it's uniform around the world. It's about 1 in 300 to 1 in 400. So I beg your pardon, it's about 3 to 4 per thousand. So it's about 1 in 250 to 1 in 300. So my maths will be wrong. But the chances of having dizygotic twins, now that varies. And it varies by nationality. So it differs, for example, Japan is reasonably low, Nigeria is reasonably high. But in the UK, we're seeing more and more dizygotic twins for a couple of reasons. One is the older the mother is, the more likely the mother is to release 2 eggs. As you know, there are many sperms. So if you're releasing more eggs, you're more likely to get this, to have 2 eggs fertilized. And this other thing is, as people know, reproductive fertilization technologies and things that help individuals to release more eggs to be able to have more likely to have success with IVF and other programs. So when we look at the breakdown of monozygotic versus dizygotic twins in the population, this is changing over time, not because the MZ rate's changing, but because the DZ rate is changing. And so if we were talking beforehand that when we were younger, we used to quote about 1 in 80 pregnancies would be twins, and now it's probably about 1 in 65. But there's more than one thing which is contributing to that. But in our cohort of twins here at TwinsUK, we are enriched with individuals who are monozygotic twins relative to the population. But to come back to your question about the same sex versus different sex, obviously DZ twins are no more likely to be sister or brother than any other siblings in whichever order. But you then have removed the added element of gender difference when you're trying to do the comparisons. And so it's useful with it to further, when you are studying these things, when you've got a brother and a sister pair, they're going to have very different sexual hormone profiles. As an endocrinologist, I could talk about this till the cows come home, but they're going to have very different sex hormone profiles from very early on in conception, whereas same-sex twins will have the same hormone profiles from very early on after conception. And so that does add weight to why it's more useful to look at same-sex twins when you're trying to dissect genetic susceptibility to a disease.
Great. Thanks, Emma. And I guess, given that we're talking about genetics, let's move on to the subject of genetics now. In terms of what have the twins contributed to genetics, and I think really the real contribution of the TwinsUK to genetics kind of started in the early 2000s, I guess, so 2005 onwards, thereabouts. When the Human Genome Project had been completed and the first human genome had been sequenced, and the technology had evolved that we could measure lots of genetic markers across each person's DNA for an almost affordable price. In the early days, it was still pretty expensive, but and Tim Spector and TwinsUK were really kind of at the vanguard of this, so much so that actually TwinsUK has published many hundreds of genetic studies based on the information that you twins have given us, and the measurements we've taken of you, the questionnaires you've answered. And
are you sure it's only in the hundreds? I would be surprised if it wasn't.
Probably thousands, actually.
Yeah, I was going to say it's likely to be a four-figure number.
Yeah, it probably is, because it's the seventh most used cohort across the entire world is TwinsUK. Wow. And there are some really big American huge investment cohorts that kind of lead the field. So we've done really well, and certainly at the top in the UK, and indeed, as I say, seventh in the world. So the TwinsUK have managed to help contribute to understand the genetics of many diseases. Yeah. And in particular, we've been concentrating on the relatively common diseases of aging, be it diabetes, obesity, high blood pressure, short-sightedness, long-sightedness. We've got a real, obviously, range of phenotypes of measurements that we take on the twins. And what we have discovered in this field is that for common diseases of aging in particular, there are many genes of small effect that contribute to those diseases. And so we realized that even with our thousands of twins contributing to studies, we needed to have even bigger studies. And so TwinsUK has shared your data, obviously confidentially, only the genetic data that other people can't identify you with, but essentially with our collaborators across the world, we've managed to get these huge studies together, and they've got bigger and bigger over the years, so that we can understand more and more about the genetics of common conditions. And by way of example, we from TwinsUK, we led the study across the world of over 500,000 people looking at eyesight, at myopia recently. And again, these were data from all over, but the TwinsUK, we led that study, and we were very proud. And indeed, yeah, over 500,000 people contributed to that study as well as our twins. So pretty impressive figures.
Oh, I think it really is a testament to people who are volunteering their time, their effort, and their data to help other people. Some of this information may help people who have been volunteers, but for most people, they do it entirely altruistically to help other human beings down the line, which is very laudable. I was actually thinking, Chris, when you were speaking, there have been studies which twins have led from the start, but there have also been studies that twins have come in to collaborate and help with down the line. Like when my first interaction with twins was when I was doing a study with osteoporosis, trying to determine what determines bone density, noting that osteoporosis is, until COVID, broken bones was the #1 reason why a white woman would be admitted to hospital after the age of 40 in the UK. I can't comment about other ethnic individuals because that data in the UK isn't so available, but it wasn't diabetes or breast cancer or heart disease or lung disease. It was osteoporotic fracture. Anyway, so I was working in this area, and we'd found a number of genes, and we wanted to see in our cohort, did these results replicate with twins? So we would ask twins for summary data that would say, you know, did this same signal that we saw in cohort in Australia, did this correlate with what was being seen in the twins? And the answer was, yes, it was. And that meant that it was much more likely to be a real thing and not just a chance finding. And so although there's been things twins have led, there's also been things that twins have collaborated with to help support. And I think both of those have contributed to why twins has been so important to the genetics environment worldwide.
Yeah. Exactly. And a lot of these studies that we're talking about are what we call genome-wide association studies, where we have we place markers all along the DNA, nowadays 1 million of them or even more.
Oh, and absolutely. Yes. And being able to not just place the markers, but work out the ones which are nearby. So most studies now will look at about 4 million of these individual variation prints for every person in the study, which gives you an idea of how much computing is necessary.
Yes, big computers required. But also with the whole genome sequencing, TwinsUK participants were among the first to get whole genome sequencing, which is an even bigger project as part of something that was called the UK 10K. So the UK measured 10,000 genomes from around the UK. A lot of them were from cohorts with specific diseases, but actually they wanted the healthy population. So they chose 2,000 of our twin samples. And again, it was a groundbreaking study that was published in very high impact journals and has been, again, those data, that information that was generated from that study has been, again, being used by probably now thousands of other studies across the world. So that again, as twins, it's just, you know, was again at the forefront of genetics in the UK.
Yeah, again, I was going to say, again, even before I joined TwinsUK, that data was useful for us. For example, if we were sequencing a cohort and we wanted to know whether what we were seeing was specific to the disease or was just background population variation that was seen commonly, the contribution of people like the TwinsUK who were sequenced to forming a common database of human variation was incredibly valuable, because then you could make meaningful comment as to whether what you were seeing was as common in your group as in the general population, or was unique to your disease group compared to the general population. And the UK 10K data has informed, it's not just looking at the sequence data, it's then being able to inform that that imputation of other genetic information. It's incredibly valuable genetic resource for the world.
Yeah, absolutely.
Yeah.
And we're privileged to work on it. So I guess we've only got a few minutes left of our webinar. So maybe we just think about the future, Emma. Where is the future of genetics within our twins and beyond? Where do you see that going? How are we going to use all this amazing genetic information that TwinsUK and others have generated? Where do we take it from here?
Well, I'm going to talk about this from a translational perspective in terms of how is this going to go forward to helping humans in 2022 to 2032. And I was thinking that within the next 10 years, I think that there are 3 areas that we will see dramatic changes with genetics coming into common clinical practice at a practice near you. And that's cancer, newborn screening, and predictive testing, or what's sometimes called polygenic risk scores. With cancer, most people have thought about cancer in the past that, you know, if you have cancer of the lung, you'll have a defined chemotherapy regimen or a defined radiotherapy regimen, and it'll be one size fits all. But what we're going to be doing much more now is sequencing the tumor and targeting the modality of treatment, the choice of chemotherapy drugs, according to the mutations which are actually in the tumor. So we're going to see personalized cancer treatment with the hope that this will both improve tumor responsiveness and effectiveness of drugs and reduce side effects by not giving people medicines which aren't going to do them any benefit, but just make them crook. So I think we're going to see personalized cancer treatment through to the genetics through cancer genetics. The second thing I think we're going to see is newborn sequencing, where there are many diseases where if you can start treatment really early within the first few years of life, it improves outcome, not just survival, although that too, but also disability and burden of disease. And we're going to see newborn sequencing coming in so that preventable illnesses which are prevented by change or by early introduction of drugs will happen at that early stage. For those of us who are perhaps a little bit older, what I think we're going to see coming in is the use of the information we get from the genome-wide association studies that you mentioned, Chris. We have all of this information and we can build them into risk scores for things like drug reactions, or for things like what's the chance that you're going to get a disease and when would it be worth starting treatment earlier. We already can do this, for example, with cardiovascular disease. We know who's at a high risk of cardiovascular disease at an early age. And we could step in early, say with statins or more stringent blood pressure control, and make a difference to the chance of that person to go on to get a heart attack. And this is that we can do this now. We can do this also in my area. Who's at risk of getting a fracture and who's not at risk of getting a fracture? We can do this, ladies, for cervical cancer. We can predict who's likely to go on to develop cervical cancer. And if you are somebody who has a high genetic risk, then you should be screened very carefully with pap smears, plus the typing for the human papillomavirus. But if you're at low risk, once you've been vaccinated, you may not need another pap smear test ever. Now, wouldn't that be great? Yes. So you can actually predict with using genetics, using negative predictive value, who doesn't need to have a pap smear every few years. And I think we'd all agree that's a very good use of genetics. Chris, I'm going to suggest that you might like to talk about a different benefit from genetics.
Emma, that's lovely. You've covered it beautifully. And I think I agree with you. I think, helping us concentrate who needs screening and who doesn't is going to be a really useful consequence of the genetic revolution.
Yeah, Chris, could I just add one more comment just to finish our webinar? In the course of my time when I was doing my PhD, I extracted a lot of DNA, and I wanted to share with our twins the reason that why Chris and I chose this background. We had a choice of a couple of backgrounds, and the reason we chose this is that when you extract DNA, after you've been doing this for it takes about 24 hours usually, and you've spun out, you've released the DNA from the white blood cells, you've spun down things, you've kept the debris at the bottom, you've used a proteinase key to release the DNA from the proteins of which it's wrapped around. You then clean it, and then you take that and you add that to 100% alcohol, and at that point, the DNA precipitates. You keep the 100% alcohol in the freezer, like vodka, and you get it out and you precipitate. And when it precipitates out, it looks like strands. This is exactly what it looks like. And you spin it in the tube and you can see the DNA like a double helix, like what it is at a microscopic, at a molecular level. You can see this spinning in the tube when you add the 100% alcohol. So I wanted to say, just to share with twins, this is what DNA looks like when you actually precipitate it out, when you're extracting it from the blood samples that you give when you come into your twins visits.
That's a very nice thought, Emma, and a great end to our webinar. So hopefully we've explained a little bit about genetics to you. Hopefully you'll be able to come to the TwinsUK website, just search TwinsUK on your search engine and you'll find it, where there is more information. And once again, thank you all for sharing your information, your bodies, your blood samples, and so much else for us. And Emma's alarm is going off. We look forward to seeing you at all the other webinars on our website. And in the meantime, thanks for joining both Emma and me.
Thank you very much. Cheers. Goodbye.
Cheerio. Bye.